So here is a theory that we're going to talk about called the endosymbiotic theory. And this is basically the idea that eukaryotic cells, as we know them now, at some point in their history engulfed a free living prokaryotic cell that became incorporated into the cell, meaning that some bacteria was floating around and it became engulfed by the eukaryotic cell through endosymbiosis, and it didn't let go of it and somehow found a way to replicate it, make copies of it, and actually use its function as part of the cell function and inner workings. And scientists have several different modes of evidence that they use to point to this idea. And one of them is this idea that chloroplasts and mitochondria have a double membrane on the outside. Most everything else has just a symbol, have that single lipid bilayer. I mean, it's a bilayer, but it's still just a single lipid bilayer. Well, the chloroplasts and the mitochondria actually have a double bilayer. Okay. And basically, this is the theory for how it created that one bacteria went in. And what happens when a cell completes endosymbiosis is that it basically wraps whatever it's engulfing in a membrane. Okay. And so you can see how this bacteria would have already had its own membrane. And when it got incorporated into the cell, it created another membrane for it. And so now the chloroplasts have a double membrane, and they still do to this very day. Mitochondria are the same way. So the theory is that it was some sort of bacteria that could do photosynthesis, and it became incorporated into a eukaryotic cell. And now eukaryotic cells, the ones that are, in plants and photosynthetic organisms, can do photosynthesis because of this. The other piece of evidence that they point to is the fact that both mitochondria and chloroplasts have their own DNA that's separate from DNA in the rest of the body. So even you have different DNA in your mitochondria. And the reason that they think it was bacterial is because in mitochondria, it's circular DNA, just like it tends to be in a bacteria. And so they can test that, and it'll be different than your own bacteria, your own DNA. If you watch any crime dramas or anything like that, lots of times I've seen this used in a couple of different ones where they've tested somebody's mitochondrial DNA to solve a case or something like that because they were confused by the fact that somebody had two kinds of DNA. So that has been used in Hollywood. But this is the whole theory that it stems from, this idea that it was once bacteria that had its own DNA. And when it became incorporated into a eukaryotic cell, it maintained that DNA. But then, of course, the eukaryotic cell had already had its own DNA. So that's just kind of a one idea of how eukaryotic cells became more and more complex.